I'm Assistant Provost. I'm welcoming you on behalf of Provost Griffith here to this last. There will be 2 more lectures, but these are in a new sort of, not really a regular series, which has been called the Provost Distinguished Scholars Lectures. Quite a mouthful. And we will have, these lectures are for scholars and artists, educators from outside of York College. And we will have 2 lectures there. One is on modern day slavery by a professor from Harvard University. And I'm not sure about the exact dates right now. May 7th. Wednesday. May 7th, I believe. Or May 6th. May 6th, I'm sorry. At 6. Wednesday. On Wednesday, okay. And the other one is a week later, I believe, by a professor from NYU. It's on private versus societal responsibility when it comes to nutrition and food. Today, however, we have a lecture on MRI and the brain's response. And I think actually the lecture goes beyond that title, if I recall correctly, with phantom limbs. And so I think I read that somewhere in the description. And it reminded me of a film that I'd seen recently, a very scary film actually, which is based on a true story. It's the story of the former editor of the French fashion magazine Elle, who suffered a devastating what is it? Stroke. Stroke, yes, a stroke, which left him his entire body paralyzed, meaning everything, and including. I think the only things he was able to move were the eyelids. A very scary movie, very scary. And the film is about the book that he actually wrote after the event, after he had the stroke, how he copes with the situation that he's in. Whereas the only way to communicate is by having somebody very patiently working with him, reciting the alphabet, and for him to blink with his eye when the letter that he intends to communicate is being said. And based on that very time-consuming method of communicating, he wrote this book about his experience, and the film was made of that. And when we came to planning this lecture, I was reminded of that. And so I think it's fair to say that some of the most fascinating and most and scariest organs of the human body is the brain. And if we can derive from that some of the most fascinating, most scariest research at York College is being done by Professor Duncan. Then you hopefully I'm sure you will be in for a treat by Professor Duncan, who will now be introduced by the chair of the Department of Behavioral Studies, Professor Mayerovitz. Thank you very much, and thank you for being here.
I won't take too much time away from the lecture, but given Dr. Hankey's introduction, I do want to reassure everyone, given that you mentioned it was going to be scary, we are not, Professor Duncan is not going to do that to anybody here in the room to demonstrate, so you will keep the back of your head, to reassure you. Okay, it's my very great pleasure to introduce one of our newest faculty members in psychology, Dr. Robert O. Duncan. Dr. Duncan received his bachelor's, master's, and PhD degrees in psychology from the University of California, San Diego. And he came to us after working several years as a researcher at the Salk Institute, a very prestigious research institute also in San Diego, where he did research on the physiological mechanisms of visually guided behavior, and also worked to develop new MRI techniques, fMRI techniques, which he will explain to you. I cannot begin to study nerve activity, blood flow in the brain. And this is a very cutting-edge area of research, so you're getting a glimpse at something very new, very up-and-coming in psychology and basic science. And it's a place where sort of the basic research, basic understanding of the brain and the nervous system, new scientific techniques to study the brain, come together with practical implications for treating illness. So it's very important work. We're not 100% sure why Professor Duncan chose to leave beautiful sunny California to come to your college. When we got the application, we thought maybe he figured it was the other Jamaica with the sunshine and the beaches. But he came here, he saw, and we're delighted to have him. I don't want to take any more time away from Dr. Duncan's talk, so I'll turn it over to him.
Thank you very much. Thank you, Holger. Thank you, Debbie, for the very nice introduction. I'd also like to thank the Provost for organizing this lecture series, and everybody who helped, and the selection committee, and the chair, Dr. Laura Fishburne. Okay, get that right. Can we focus this a little bit? Or is it me? Is it? Okay, great. Then we're ready to roll. I just have some kind of low contrast images. I want to make sure you get the prettiest show you possibly can. Okay, you're probably right, and I'm just sitting too close. But anyway, so I'm going to do what I tell all my students never do in a talk. I have this loaded with 100 slides, and I'm ready to go. You can't stop me, certainly for clarification, but it's going to roll more like a movie. I will assume that the audience is infinitely intelligent, but also infinitely ignorant. So there are some things, apologies to the learned scholars in here and my students who know about neurons and whatnot. I will be doing some basic, not only neuroanatomy, but also some basic MRI techniques. We'll talk about some curious tales of brain injury, basic anatomy and physiology, some MRI methodology, and then the interesting stuff, which I think is the interesting stuff, is using these tools to assess individual variability, and then beyond that, applying what we learned about measuring individual variability using MRI to clinical populations, assessing brain changes associated with eye disease, which is a very subtle and difficult thing to do. And the reason we're looking at eye disease is to develop an understanding of the disease, of course, and to improve diagnoses, develop new therapies, but most importantly for me, it's how to learn how the brain responds to injury. Now, this is, of course, work in progress, so I don't have the answer. There's not going to be a big grand finale to this, but we'll start off with the course and work to define. The course, Curious Tales of Brain Injury. And I want to show you this because I want to show you the main goal in psychology is really to sort of explain psychological phenomenon using physiology. And we want to establish strong correlations between the two. And you'll find no stronger correlation, sorry, I should say changes in behavior should predict and covary with changes in physiology. And for the next cases, you'll see relationship is very easy to establish. You have profound changes in behavior that are accompanied by profound changes in physiology. And that's kind of the first step when you're doing something like applying MRI to any disease. So this is a case of neglect. This patient has had a stroke that has knocked out their right hemisphere, and so they are blind to everything in the left visual world. Okay, they have right parietal lobe damage, that's on the top of your head. They have an inability to allocate attention to the left visual field, but they can actually see there. They were given two tasks. At the top, you'll see a task where they were presented with all the black lines, and they were asked to cross out every one of the black lines with a red line, with a pen. And they could do this, but only for half of the visual field. If they're asked to replicate this drawing of a house, they'll only do so for the right half. They're visually unaware of what's going on in the left part of the world. Now you think, well, they're just blind. Maybe they're missing the hardware in the eye and they lack the ability to see, but it's an attentional disease. And you can restore that by giving them what's called a caloric. I'll pronounce that properly. It's cold water that's injected into the ear of the healthy hemisphere. And what happens is it temporarily stuns that hemisphere and sort of cripples that hemisphere for a moment and allows the natural balance between the two hemispheres to be restored. And then suddenly, the patient can see into the hemisphere that it was, into the hemifield where they could previously not see, and they could complete color net lines. Unfortunately, it's a temporary fix. But what it shows us is that neglect is caused by competition between the healthy and damaged hemisphere, and it's sort of a disease or disorder of attentional state. So here we have an obvious behavioral problem that's coupled with a very dramatic injury to the brain. Another curious story is that of blind sight, which has gotten a lot of press lately. Human blind sight's patients truly believe that they can't see. If you ask them, are they blind, they believe they're blind. Yet they actually perform a better than chance on a lot of behavioral tasks. And that's a result of having severe damage or almost complete removal of the primary visual cortex. This is the first stop that your neuron, neurons from the eye makes when it hits the brain. There's one intermediate stop, but the first stop of real consequence is in the primary visual cortex. I'm going to show you, now this is a monkey that has had its, let's hope the audio works. This is a monkey that has actually had its primary visual cortex removed, and you see it's navigating this complex environment. It's not really bumping into very many things. You'll see it bumps into something here, boom, but it can continue and it can navigate this despite the fact that the monkey is actually blind. A human with this sort of disease believes they're blind. They're afraid to navigate their environment, but if you put them in a scenario like this, you'll find that they actually don't bump into anything. In the second half of this film, you'll show a monkey that's, if you place pellets in front of the monkey and they just appear, it won't be able to find them. But if you roll them out one at a time and it's moving, then it can see it, even though it's not aware it can see it. It can find the pellet and get the reward. Okay. So again, humans and monkeys perform better than chance. And the idea is that perhaps there's a secondary pathway. We have the cortical pathway, which goes directly from the eye to the back of the head. And that's normally what we use for everyday perception. But there are a bunch of subcortical pathways that go near the brain stem, and they're responsible for things like orienting your eye movements, knowing where you're out in the world, and perhaps these subcortical pathways are sustaining some kind of primitive vision, even though you might not have conscious awareness of vision. And this begs the question, is primary visual cortex necessary for consciousness? Here these monkeys don't have primary visual cortex, and therefore they're not consciously aware of what they're doing. So it might be a fundamental substrate in thinking. Another curious tale comes from studies of prosopagnosia and visual agnosia, and these patients have a very unique type of blindness. If you show them a picture like this one at the right, patients with prosopagnosia will not be able to recognize that this is a face, but they will be able to recognize that it's composed of fruits and vegetables. They can see the objects, but they can't see the face. Patients with damage to fusiform face area underneath the bottom part of the brain have the exact opposite problem. They can't see the face, they can only see the fruits. There are other patients with visual agnosia who can only see the face but can't see the objects. Distinct brain regions resulting in very different and dramatic problems with behavior. An associative visual agnosia. You can give this person a model to copy. Here they were given an anchor, far left. And if they're asked to copy the anchor that was placed in front of them, they can do a pretty good job of it. That's the second panel, B. But if you ask them to draw an anchor from memory, you get these results, C and D. Same patient. Somehow there's a dissociation between their ability to see and their conscious ability to produce drawings. They clearly have the manual dexterity for it, they have the visual system for it. They can see, they can move their hands, yet there's something in between that's not working right with these patients. If you take these last three disorders, like prosopagnosia and visual agnosia, and you lump them all together, you might get something that is as complicated as the Capgras delusion. Now, the Capgras delusion is a very interesting case. A person will not be able to recognize specific people in their lives. They've had brain damage, and they will say, you know, doctor, I see this patient. Doctor, I go home and I see my mother. She looks just like my mother. She cooks like my mother. She talks like my mother. But it's actually not my mother. It's an imposter. a specific person has sort of been removed from that person's life. I'm just going to show you, I'd like to show you in the patient's ward. This is from a film called Secrets of the Mind, and the doctor you'll see here is Dr. Vilayana Ramachandran at UCSD.
You know, I'm sure you would like to meet this guy. He's so much like you, but he drives better. He doesn't go so fast.
It can look identical to him, exactly like him, but it's not him.
Say, this woman, doctor, she looks exactly like my mother, but in fact, she's not my mother. She's an imposter. She's some other woman pretending to be my mother.
The injury to David's brain had brought on a very rare condition called the Capgras delusion.
I was cooking dinner, and he probably didn't like the food that night.
Okay.
And he said, you know, the lady who comes in the morning, she cooks much better than you.
Okay.
It's that lady, I like that lady very much.
Okay.
But the lady was me, of course, all the time.
David was also convinced that his father was an imposter.
He would say to his dad, you know, I'm sure you would like to meet this guy. He's so much like you, but he drives better. He doesn't go so fast.
It can look identical to him, exactly like him, but it's not him.
So imagine being that person's parents. The trick that they actually used to connect with their son was by calling him on the phone, and he would recognize them on the phone, and they could have conversations on the phone. But, you know, or they would walk him outside the house, walk him back in through the front door, and then he would feel, oh, I recognize my mom and my dad again. Eventually, it did go away. Last tale, temporal lobe epilepsy. This is a patient who has severe epilepsy that affects a region called the amygdala, which is responsible for emotion in the brain. And normally, the amygdala's job is to sort of stamp incoming data as being biologically relevant or not, a memory worth having. Data comes in, the amygdala says, this is important to you, stamps it, and then you store it. Now, this patient's amygdala is constantly hyperstimulated by the epileptic seizures, so he assigns a lot of emotional significance to everything in the world, arbitrary objects, cups, chairs, podiums, speakers. And he believes that he's having a religious experience and the world is completely beautiful.
John endures.
I've been in so much pain that I'd rather be shot to death, dude, or just whipped to death.
But
Whipped
also
to death.
joy?
Yeah, I've been in so much joy that I would rather be left alone, man. Take everything away and just let me sit there and have that much joy. I feel like I can float and stuff sometimes, you know?
Okay.
It's like the best.
There were times where he would have 7 or 8 grand mal seizures in a day. He would never come back to this reality during that time. I have looked in his eyes in those times, and I have seen a cry for help.
No, I mean, you guys, that's the thing, though. A lot of other people can just walk around and see the beauty of the world. I can... Sorry. It's not as beautiful.
So I could show you the brain scans of these people. It would be quite obvious. They're missing large portions of their brain due to traumatic accident or some kind of epileptic seizure. That's not really a mystery. But what is a mystery and interesting, I think, to note is that there is a clear separation of awareness and the true physical nature of the world. These people have an atypical representation or perception of the sensory stimulation that's coming into them. And again, of course, profound changes in behavior can be explained by profound alterations in physiology. Which leads me to my thoughts on American Idol. Perhaps the part of the brain that's responsible for singing is located very, very far away from the part that is responsible that you know you can sing. I will be recruiting subjects soon for this one. Okay, a very, very quick tour of anatomy and physiology. Your eye is connected to your brain. That's about as much as you need to know for this. The purpose of the eye is to focus light to a point at the back of the eye called the fovea. The fovea pit has a very high concentration of photoreceptors which convert light energy into neural impulses. And those nerves send projections out through the optic nerve to the brain. Here's a close-up of that. You have photoreceptors at the far left again, responsible for converting light energy into neural impulses. They pass through a meshwork of relay cells and then eventually reach the ganglion cells. When I talked about glaucoma, these are the cells that are the most affected in glaucoma, and those projections get sent to the brain. There are neurons in your brain. They talk to one another. That's important. And here's again a representation of the cortex and the right hemisphere. Just to reinforce this, the right hemisphere, the primary visual cortex, is responsible for the left hemifield in visual space, and vice versa. I'll skip over that. Most of the cell bodies in your brain reside in the gray matter. That's this dark region out here. And the white matter is mostly composed of axonal projections that connect these regions. And the reason your brain's all wrinkly is to fit a very large surface area into a very small box. So how do we actually measure the brain? When you think of MRI, magnetic resonance imaging, you typically think of a static snapshot, a picture that's used to diagnose some type of brain defect. That's what you see at left. It's high resolution. Standard is probably 1 by 1 by 1 voxels. We don't say pixels, we say voxels. They're volume pixels. But when you're interested in brain function and research, we use functional magnetic resonance imaging, which is a way of inferring neural activity by looking at blood flow. So it's slightly lower resolution. Standard is around 3 millimeter voxels, where in the traditional anatomical MRI, you take one picture. In fMRI, you'll take several pictures over time, usually around 5 minutes. And this method is called the blood oxygen level dependent signal. We look for changes in hemoglobin. Changes in blood that's been used and blood that hasn't been used have different magnetic properties, and you can detect that using the fMRI machine. So increases in neural activity result in increased demand for blood oxygen, which results in an increase in the fMRI signal. A classic MRI experiment would go something like this. You have a subject sitting in the bore of the scanner, and they'll be looking through a projector at a central fixation target like that red dot. And then you present them with some type of flickering stimulus. In this case, it's a full field checkerboard, and that oscillates every 8 hertz. And you turn that entire stimulus on or off every 20 seconds or so. So they're getting massive stimulation, and then it's nothing, and then massive stimulation and nothing. Then you grab some slices at the back of the brain. Here's one of those slices. Now, as though you're looking at your patient, and I'm going to look at 1 voxel for that patient. And you can see as we turn the stimulus on and off, there's an increase and decrease in the pixel intensity. It's just brightness. We're basically doing image analysis. And that goes on and off for the duration of the scan. You can fit that with a sine wave. And the goal in MRI is to compare the fMRI signal's stimulus time, compare the fMRI signal to the stimulus timing. So we say that this response is phase locked to the presentation of the stimulus, and then you can look at the amplitude, the magnitude of the strength that's phase locked to the timing of the stimulus. What you'll see in this talk and in many MRI talks is one paradigm. The most common paradigm is called the block design. You have condition 1, you want to compare it to condition 2. Condition 1, you take a bunch of functional images, and then in about 20 seconds later, you show stimulus 2, collect more images. That goes on for about 5 minutes, and then you also take multiple scans, maybe multiple days. Then you look at one particular region of interest. In our case, we're interested in primary visual cortex. And you can plot the signal change for that particular region only. You don't want to look at the whole brain because you're going to get other arbitrary regions that oscillate with the stimulus, maybe because of attention or something like that. Maybe they hear the sound of the scanner. You'll expect activity everywhere in the brain. So you have to sort of go in there with an a priori knowledge about what you're looking for. And then you can sort of average and get condition 1 versus condition 2. You can average that entire signal. And even project that back onto the anatomical image as a statistical map. Okay, I'm going to warn you, this is only going to work once because it's a kind of quirky movie. Another way to look at the data is on a flattened representation of the cortex. So imagine you're cutting the back end of a basketball off, and you have to cut slices into it, and you flatten it completely. That's what we do computationally with MRI. So take a look at the left image. First, you inflate the brain. Let's going to be fussy. There we go. Yep, it's going to be fussy. All right, so let's try the right one. And then you flatten it out. Right. Yes, it's tricky. It's all done with magic and mirrors. This is, so these are two hemispheres. The back of the head has been cut off. The left and right hemisphere has been flattened. And this is the visual cortex, primary visual cortex for one patient. And that patient is looking at an expanding ring here presented at the bottom. You can see this traveling wave of activity that goes from the central representation of vision all the way out to the peripheral representation. These colored regions are used to denote different visual areas, primary visual cortex, V2 and V3, which have different processing jobs. It's a standard stimulus we do, or cartoon representation of it. And this is a rotating wedge, another standard stimulus. Now, you'll notice here that when the stimulus is moving throughout the right portion of visual space here, the left hemisphere becomes activated and vice versa. You'll also note there's kind of reversals in phase as it goes from the bottom to the top. You'll notice that this wave of activity comes from the top to the bottom. And then it will reverse its phase at the border. We use this phase reversal to determine the borders of visual areas. Here's a more accurate representation of what the stimulus looks like. It's that expanding ring and the rotating wedge, but it's flickering like you saw before. And that's how we basically obtain a retinotopic map of the world. We can now know where each specific point in the world is represented in a particular person's brain. So the next question is, do differences in perceptual ability correlate with differences in brain area? So here's a cartoon representation of a retinotopic map. If you were to grab two individual adjacent points in the primary visual cortex and ask what do those neurons see, you would see that this particular area in blue is responsible for encoding all the information that is located in this particular region near the center of vision. And it actually looks at quite a large space. Out in the periphery, you get the opposite here. So acuity, when I say acuity, I see how fine your vision is. It depends upon receptive field size. So each neuron has a small view of the world. Neurons out in the periphery of your brain have a very large view of the world, but they can't resolve much detail. Neurons nearing the center of your gaze have very small receptive fields, and they're very good for resolving detail. So if you were to look at these horizontal stripes, and you were to look directly at one of the stripes, you would have a receptive field that fits within one of those bars, and you could tell the difference between the white bars and the black bars. Now, if you were to look way off at the exit sign, for example, in this room, and you were to try and resolve the stripes, you would have some neurons that were responsible for that portion of visual space out in the periphery with a much larger receptive field, and all the black and white bars would fall in the confines of that receptive field, and you would not be able to resolve the detail. Another way of looking at it, any small region of this image, you can take a look at the image yourself and inspect it. Obviously, it's a quite curious, impossible figure. It takes a little while for you to find it, but it's not hidden, right? The legs are quite clearly not consistent with the empty spaces in between the legs. Any small region of this image makes sense, but the whole image does not make sense. And partially that can be explained by receptive fields. So if you're looking at one portion of the image, like the animal's eye, receptive fields that are nearest to the center of gaze are very small, so you can resolve that detail. However, if you're looking at the eye, receptive fields way out in the periphery are quite large, and they can't really discriminate the differences between the legs and the space. So you don't make sense of that. If you were to look at the leg, then locally that would make sense detail-wise. So neurons with large eccentric receptive fields don't provide us with much information about detail. You can look at that in this left plot here as eccentricity. The distance from the center of the gaze increases. Your visual acuity goes down. Your ability to discriminate fine detail goes down. If you're to look at this center dot here, you'll notice you'll be able to read the letters that are way out in the periphery. But if you look at the letter A, for example, and try and read the letters that are at the center near the dot. You won't be able to do that. The letters have been scaled with respect to receptive field size and eccentricity so that when you look at the dot, you can actually read them. So related to this is the notion of cortical magnification. The center of your gaze is represented by a larger proportion of the cortex. So this is a famous experiment by Roger Tuttle where they put a monkey, they stimulated a monkey using a flickering checkerboard stimulus, and the monkey was fixating at the center here. And then they processed the monkeys, they sacrificed the monkey and they processed it using radioactivity, 2-deoxyglucose, and you could map basically that grid on the brain. And what you'll notice here is this is the center of the gaze, and it's represented by this large proportion, this large area out here, even though the bars are much more narrowly confined. You have a very small difference between the inner ring and the outer ring, but it's still represented by a very large space here at the center of cortical magnification. Applying that as data, you see increases in eccentricity results on the panel at the left, in decreased cortical magnification, and at right, increases in eccentricity result in larger receptive field sizes. So going back to individual differences, we have some specific research questions to ask. Acuity and cortical magnification depend on eccentricity, and does acuity correlate with cortical magnification? Not only that, but do individual differences in acuity correlate with cortical magnification? And to explore these questions, Jeff Boynton at the Salk Institute and I, now at University of Washington, put human subjects in an MRI scanner and measured their acuity. So outside of the scanner, humans were asked to look at the central fixation target, and then we presented them with two vertical lines. And the vertical lines would either be perfectly collinear or they'd be slightly off, like in this picture. And you could adjust that difficulty and measure how well they do. This stimulus is presented all over the visual field, and you get basically a broad survey of how well they see everywhere in visual space. Then you bring it into the MRI scanner, and then you present them with a flickering ring. And that ring could be at various eccentricities. It could be very close here, as in red, or it could be very far out, as in blue. So a very large ring or a very small ring. This is the flattened hemisphere for one subject. The bright pixels indicate regions of activity, and you can fit that with the line here. So this line is a fit. The stimulus that was shown was this 1.5 degree ring here. Then as we show larger and larger rings, you can see that the activity moves further and further away from the representation at the center of gaze. We can also measure the vertical meridian and the superior and the horizontal meridian, that is the line that runs down this way and the line that runs down that way. And you glue all those together and you get a template, and that template describes that particular person's representation of visual space. Here are a bunch of templates for a bunch of subjects. This is me. You'll notice that some visual cortices, the representations are larger than others. Some have asymmetries, this particular patient here. These are all perfectly seen subjects. They have normal vision. There's no defects in them. The question is, do they have difference in their visual ability? So what we did is we plot their visual acuity as measured in the behavioral test against this measurement of cortical magnification, meaning how much of the brain is dedicated to a particular part of visual space. And there's a correlation between the two. We showed two stimuli. The one that I showed you was called the vernier stimuli. Those are the two lines that are slightly off axis. So as you get further away from the center of gaze, your performance decreases, and cortical magnification is inversely scaled with that. So it appears that for individuals, cortical magnification is correlated with your visual ability. And this is all the subjects plotted as individuals. That was the group data. So next question is, does this relationship exist for all sensory modalities? This is a cross-section of your skin. You have various types of receptors, 4 receptors, Meissner's corpuscles, Pacinian corpuscles. They're all pressure detectors. Some are connected to the hair follicles, some are not. But we have a very sensitive fingertips. We have a high degree of sensitivity in the fingertips and in the lips. And that's usually what we use to explore the world, and that's where we would expect to find the most cortical magnification for the part of the brain that is innervated, the fingertips, I should say. This is the long and boring path. From the periphery, it goes to the spinal cord and up into the primary somatosensory cortex, which is what I'll be looking at. If you were to look at a patient head-on and map out the somatotopic representation of your body, you would see that certain regions have a much larger representation in the brain. Right here, the hands take up a huge portion of the somatosensory cortex, as does the face. Other parts, like the knees, not so much big. We don't use our knees for exploring the world. Funny world if we did. Now, just to give you a notion of how this works, I want to show you another critter. This is the star-faced mole. The star-faced mole lives much of his or her life underground, and this is magnified. The mole is about, let's say 6 inches large, not 30 feet in height. And the first thing I think, obviously the nose of the star-faced mole is the most curious thing, but the second curious thing are the giant claws for digging. The mole spends all its time underground digging, and looking for food. Well, when its hands are occupied with these giant digging claws, it doesn't have the fine tactile acuity necessary to resolve a worm from dirt, from leaves, and so on and so forth. So it has evolved to develop these large finger-like appendages on its nose, I believe about 12 in all. This is a representation, a cartoon representation, of the nose appendages and the hands and the rest of the body for that mole. Quite different from ours. We have a large hand representation, a small nose representation, but it's quite biologically relevant to that animal to have that. And it suits it quite well. I believe it's actually the fastest eater, the fastest known eater in mammals at least. So what we did was we measured people's tactile acuity and then, of course, their brain response on a task where they had to distinguish 3 bumps on a surface that was etched in using acid. So they have 3 bumps, and the job was to determine whether the middle bump was offset or not, or was it collinear. And the task gets progressively difficult as they get better and better, and so on and so forth. Of course, they can't see this. So they're blindfolded, and they're just asked to place their finger down, and they can feel it and make that judgment. The fingers are labeled D2, 3, 4, and 5. D2 is your index finger. D5 is your pinky finger. As you move from the index finger out to the pinky finger, you'll notice a dramatic increase in threshold. That means you get worse and worse. We don't use our pinky fingers as much for exploring as we do, say, our thumbs and index fingers. So we have the highest, very, very noticeable improvement in tactile acuity using the index finger relative to the pinky fingers and the others. Here is the fMRI responses for one subject. So what we did was we put subjects in the scanner, and then we stroked one of their fingers for 20 seconds on, and then nothing. 20 seconds on, and then nothing. And then you can plot that activity in the brain. This is the flattened representation of somatosensory cortex. This bright pixels indicate a region of activity, and we fit that with a line. That's the index finger here, middle finger in the second panel, third panel, ring, and then the little finger out here. And again, just like in visual cortex, we could get a template for that one subject. Just right here in this 3D representation of the brain, this is the actual gyrus sticking out in somatosensory cortex, and you can see the red region is the index finger representation. It's much larger than for the little finger, which is in blue. Plotting those, this is for all subjects in all hemisphere. You can see there's a clear and strong correlation between the psychophysical measurements of tactile acuity and the measurement of cortical magnification, or the area dedicated to that finger. So visual acuity depends on the area of brain devoted to particular eccentricity in vision, and tactile acuity depends on the area of the brain devoted to a particular region of the skin. And they seem to co-vary with cortical magnification between individuals. So the thing to take away of this is the first steps. If we can measure this type of sensitivity between individuals, then we'll have the ability to look at diseases, hopefully. fMRI methods are sensitive enough to detect these subtle variations. So the next step was to look at eye disease, start to develop clinical applications of this technology. I'm going to skip over a bit of plasticity, but the idea of plasticity is that when you learn something, the connections between the neurons in your brain change anytime you learn something. But also when you have an injury, let's say you have an arm removed, the brain must reorganize in response to that injury. It no longer has input, so it has to change. These neurons are sitting doing nothing in the brain, so now they take on new roles and responsibilities. That being said, I think I can skip over. This is a case of a young girl who was having seizures so bad that she had to have half of her brain removed. If you do that before a critical period, the entire brain can reorganize. And you'll note this girl is actually, despite the fact that she holds her left hand a little awkwardly, normal girl in every other sense of the world.
We are beginning to harness the brain's incredible ability to invent itself, then reinvent itself throughout life. This girl is a testament to the amazing resilience of the human brain. Young Jody Miller leads an idyllic life as a 9-year-old girl. You would never guess that she has undergone some of the most drastic surgery imaginable. Jody's first 3 years were textbook normal.
Happy birthday, dear Jody.
Then, about 6 weeks after her 3rd birthday, a storm of epileptic seizures took control of her brain.
She couldn't use her left arm hardly at all. She could barely use the left leg, seizing a good deal of the time, multiple types of seizures.
Quite clearly dramatic. But I think the most interesting thing is how normal she appears. This is a different patient, but this is the type of damage you would see. The very white regions out here are actually parts of the brain that are missing are now filled with cerebral spinal fluid. Here, I'm going to skip over this one. So there are cases when adults have had amputation for various regions and they have phantom limbs, which I'm sure many of you are familiar with. Phantom limb is the perception that the missing limb is still there. They'll either have some type of sensation or pain, chronic pain actually. And the reason is actually, it baffled people for a while, and sometimes you would re-amputate, maybe it was the surgery that went wrong, but eventually they figured out that it's actually the brain. It's maintaining a representation of a limb that does not exist. The severe pain can be very, very crippling and occupies a lot of their mind. You can have phantom eyes, phantom uteruses. What else do we have? Limbs. Basically anything that can be removed can cause a phantom. In the cases of pain, the idea is that the subject has learned pain. You remove the arm. If the arm was in pain before the amputation, the brain has sort of learned to be in pain. Amputate the arm, and now that representation of pain still exists in the brain. So one of the ideas is to try and re-teach the brain that it's not in pain. And Dr. Ramachandran at UCSD has developed a very simple $2 solution to this. It's called the mirror box. This is a cartoon, but imagine this patient has actually had, I say this is Dr. Ramachandran himself, but he's not really missing his left arm to my knowledge. He's just got it pulled up into his sleeve. But imagine this were a patient who is missing his left hand. You place a mirror between the left and the right hand, and the patient looks at the mirror representation as they're moving both hands in synchrony. So the brain command is saying, I'm moving both of my hands in synchrony, and then it looks at this mirror and it says, oh, I have 2 healthy hands. It's getting visual feedback. The patient is getting visual feedback, and they eventually, even though they know their left hand is clearly missing, they're not crazy, they're getting visual stimulation, and the brain eventually can relearn to not be in pain. And this therapy, while it's not a cure-all, patients report less pain, and some have reported their pain not coming back. Here's a more sophisticated version of that using virtual reality glasses. This patient is giving a number of tasks. You'll notice that he's getting visual feedback from the left hand only. He's asked to play with a ball, answer various questions, usually like a touchpad. But of course, he actually has no left hand. His right hand has been fit with a glove that feeds into the VR system. And this patient, again, reports having much decreased pain after that. So this is the essence of neuroplasticity, the brain relearning something. First learning something incorrectly, and then learning to readapt to the new situation with the appropriate input. So car accidents and the like take limbs very quickly. What happens if you lose peripheral input very slowly? What does the brain do then? And so what I do is I'm using glaucoma as a model to study plasticity. Glaucoma is an eye disease where you lose vision, peripheral vision, very, very slowly over a period of 20 to 40 years. It's a very insidious disease. You don't actually know you have it until you're tested or it's too late. It's actually the second leading cause of blindness worldwide. The most common cause is cause of increase in intraocular pressure. That pressure goes up, it puts pressure on the back of the optic disc where the optic nerve leaves the eye, and it starts to damage those axons and nerves. The death of the retinal ganglion cells results in an irreversible vision loss. It can't be cured, it can only be stopped at this point. The curious thing comes from patients who have glaucoma, but they don't have increased intraocular pressure. If it's a disease of pressure, then how can you have glaucoma without pressure? And that's where the brain side comes in. We're starting to look at glaucoma more as a disease like Alzheimer's or neurodegenerative disease that might have multiple contributing factors. So my job here is to understand the pathology that may lead to new diagnoses, new treatments, and to understand this disorder better. So this is a cross-section of the eye. Normally you have fluid is constantly being created in your eye, and it exits out through little vents near the iris. Occasionally, that aqueous outflow can become blocked and results in a buildup of pressure. To give you a demo of what the optics did, so as the nerves leave the back of the eye, you have what's called a blind spot. If you were to all take your thumbs and look through close your left eye, look at your left thumb, and then slowly move your right thumb out. When you get about this far out, the top of your right thumb will disappear. Keep in mind you're supposed to look at your left thumb, and you're only looking through your right eye. Anybody getting that? Okay. Yeah, a few. Both thumbs straight ahead, look through your right eye only. Look at the left thumb and slowly move the right thumb out. The top will disappear. We don't have any receptors in that particular portion of the visual world. That's where all your nerve bundles are leaving the eye. So we can't have any receptors there because there's just basically a hole in the back of the eye. That is where the damage to glaucoma occurs, at that region, where all those nerves are leaving. Here's a schematic. If you were to look straight on, this is the fundus. Here's the fovea, which is you have a high concentration of cells. This is the optic disc, where all those axons are leaving. The increased pressure causes cupping in the disc down here. And the patient slowly loses vision from the outward in, from the periphery inward. Here's another visual illusion. We're not always aware, so the point in showing this illusion is that we're not always aware, you weren't aware of the blind spot, right? The blind spot exists for all of us, yet we fill in, even if we're only looking for one eye, the brain is taking all the visual information it can, and it's making a sort of representation of the world. It glues all the pieces it has and fills in for the pieces it doesn't. I want you to fixate at this central fixation, at this crosshair. I'm going to start this movie. And eventually, you'll see a green dot traveling around the circumference. All right, people getting that? Okay, now try look at the green dot. Goes away, right? Now go back to the fixation target. What you're seeing is an illusion. Your brain is filling in for the content it has. I'm going to stop this. These purple spots are creating what's called an after image. The after image of a purple spot is a green spot. As that gap moves around, it's creating a green after image, and your brain is tracking that green after image around the circumference, and you say, oh, there clearly must be a green spot moving around. It's taking what information it has, inaccurate as it is, and filling in. That's the problem with glaucoma. We're so good at filling in things is that people don't notice it until they get it too late. So we need early diagnosis. We need to get people into clinics and look at the backs of their eyes and see what's going on. So the research objectives in this project is to determine the relationship between cortical function and perception, determine the relationship between cortical function and damage to the optic disc, measure damage throughout the visual pathway, and I'm going to look at the first one now. So we bring people into the clinic. They do a variety of standard clinical tests where they do detection of very dim stimuli in the periphery. Here you have the results of one of those tests called the standard automated perimetry test. Here's the right eye and the left eye. These big black spots mean that they couldn't see something there. So you can see that this particular patient has a relatively intact left eye, but their right eye is affected by glaucoma. If you take a picture of the back of the eye, fundus image, you'll notice that in the healthy eye, there's this thick neural retinal rim that is gone from the damaged eye. That's the effects of glaucoma. The pressure is killing off these optic nerves. If you're to look at this in 3D, these are actually 3D stereo pairs that if you can look at these in 3D, you'll notice a large amount of cupping in this one, but not that one. So this is the stimuli we showed our patients, mapping the meridians, the rotating wedges, the expanding rings, and so on and so forth. And this is one patient's flattened hemisphere. You notice this giant ring that corresponds to the arc, that flickering arc that we presented. We can fit that with a line. We can fit the vertical meridians, the horizontal meridians, all with lines, and we get a template, a much simpler template than what we used before, but a template that describes that person's visual cortex nonetheless. We have to be quick and dirty. With the patients, we only have 1 hour, so we reduce our stimulus set, but it's still accurate. Then we have a stimulus, what we call the scotoma mapping stimulus. We're interested in finding out what parts of the visual field they're blind in. So we just close, we don't close one eye. To the right eye, we present a full field flickering checkerboard. And the subjects are fixating a target over here in the corner that tells them which eye to look through. And then that stimulus flips around, and then they look through their left eye and their right eye and so on and so forth for about 5 minutes. And here is the results of that. So a reminder, this particular patient had damage in their superior nasal visual field in the right eye. So it's their right eye, the up left-hand portion of the world. The blue region here, so this is their hemisphere now mapped in color, flattened, and the template from the Retinotopy superimposed upon the data from the experiment. So this tells us where their visual cortex is. This black triangle is a projection of this entire quadrant of visual space onto the flattened cortex. So we're looking at this particular region only. The color of the pixels tell us which eye has better vision. So the blue pixels tell us that they're better at viewing when they're looking through their left eye, the undamaged eye. Remember, we were just looking at a comparison, left eye, right eye stimulation. You can quantify that. Well, let's say I think that's this patient right over here. Of course, showing my best foot first. A large fMRI signal that's statistically different from 0. If your right eye was as healthy as your left eye, you'd expect to see nothing in this test. And you can plot for various tests of visual function the difference between the change in the fMRI signal and the difference between their right and left eye visual function. So the larger the difference out here, that means they had a very big difference between their left and right eye in terms of visual function, which is also accompanied by a very large difference in the MRI signal. And this is for just other tests, other standard tests of visual function, the same patients. So the next part is to determine the relationship between cortical function and damage to the optic disc. The same idea. You bring people in, you test. Now you're not only looking at their behavioral measurements now. Before we looked at the behavioral measurements, now you're directly measuring the thickness and the health of the optic disc itself. This is usually a scanner called the GDX. So this is a picture of the back of that subject's eye. This annulus right here is where the neural retinal rim is. Yellow regions indicate thick, healthy nerve fibers. So here we have thick and healthy nerve fibers in the left eye, where they have good vision, and not so much in the right eye. You compute the mean thickness within that ring and compare the unhealthy eye to the right eye, just like we did before. The same thing, that patient's map. You notice the large difference here in the region of interest, showing that they can see better through their left eye. And again, for all patients, the same type of correlation. If you compare the fMRI signal here to the thickness and health of the retinal nerve fibers, you see you get a strong correlation between the two. So we have the ability to track these patients now over time and look at the progression of the disease. But the question is, well, what is the brain doing when the eye is damaged? It's quite possible that when we're measuring this brain function, we're measuring damaged brain. The fMRI signal can be measured damages to the brain, but it'll also be measuring reduced input because the eye itself is damaged. So another method of studying this is to not stimulate the patients, just bring them into the scanner and measure their resting blood flow in the cortex. So no visual stimulation, patients are just resting in a dark room, and we use a different technique called arterial spin labeling. This is the damage for that one particular patient. In their right eye, they have damage to the upper visual field, and their left eye is relatively normal. And this is with a separate test, a different type of test, same type of damage. And this is just a representation of the brain signal, a cartoon of that. And we compare the upper versus, in this particular case, we're comparing the upper visual fields to the lower visual field. In this patient, there's a lot of damage, and we compare that to the upper versus lower representation, or the ventral versus dorsal representation in the brain. And we find when subjects are just resting in a scanner, there's a correlation between the blood flow at rest and the damage that that patient displays in behavioral testing. So without any visual stimulation whatsoever, you can tell that the brain itself has been affected. So preliminary conclusions, changes in visual function caused by glaucoma are correlated with changes in cortical function, and changes in the optic disc structure in glaucoma are correlated with changes in cortical function. Traditional bold fMRI is a useful method for monitoring these patients over time, but you can also use ASL to confirm glaucoma and damage to the cortex. Now the next and final step is to measure damage throughout the cortical pathway. Okay, we can measure damage in the brain. Can we look at smaller structures? Now we move one step forward, and we're going to now look at the first relay between the eye and the brain. It's called the lateral genicular nucleus, and it is in the thalamus at the middle of the brain. It's a very, very small structure located here. It's laminar with different layers representing the right or the left eye, and also they have different cell types. They receive from different cell types in the eye. And then we can use arterial spin labeling again. So I'll go into that in a little more detail. Arterial spin labeling works like a tracer. You magnetically tag the blood at the neck, meaning you hit all of the hydrogen protons with a magnetic signal. It flips them all on one axis. And those labeled protons move throughout the brain, and they eventually reach the center of the brain, and you compare blood that has been tagged with blood that hasn't. So it's a non-chemical tracer. When we're looking at the LGN, we look at the left versus right hemifield. We alternate stimulation between the left and right hemifield using a flickering checkerboard. And here you can see the obvious difference in stimulation in the primary visual cortex, but also way out here in the lateral genicular nuclei, these little regions where the arrow's pointed. And if you look, the stimulus onset goes on and off, left and right, left and right. And for this patient, you can see that there is this excitotoxicity. These cells go into spasms in the eye. So the blood flow fluctuation itself may lead to retinal cell death. This is a picture at left. There's a picture straight on at the back of the eye. You can see these large arteries here, the central retinal artery, which is what most ophthalmologists see when they look at the back of the eye. But unfortunately, this is not the blood supply to the parts of the eye that are sensitive to glaucoma. if you look at the cross-section of this, the central retinal artery passes through the optic nerve and then unfolds along the fundus. But the meshwork that actually serves the back of the eye is these little buggers over here, the ciliary arteries. And you can't look at that meshwork of blood vessels by taking a picture through the retina. You have to use MRI or something that looks at it through the back of the eye. So again, we've done this now not in glaucoma patients, but in normal people with normal vision. Use arterial spin labeling. The blood is magnetically tagged at the neck. It travels up into the eye, and then we acquire the data with a single slice that's parallel to the optic nerve. Here is the image of the eye. This is the retina and the choroid, the part that nourishes the cells in the back of the eye. We've just drawn some pixels on there to tell us which part of the eye we're actually interested in looking at. This is the time course of the stimulus in red, the dotted line. And you can see the time course of the blood flow to the eye follows the time course of the stimulus. We've managed to get this working in 2 patients. Patient 1, patient 2. In this case, it looks differently. We used a fat suppression algorithm to hopefully improve the signal. We get some improvement in the signal, judged by the overall magnitude. But again, you can see that we have the ability to measure this blood flow. The problem, even though ASL fMRI is sensitive enough to measure ocular blood flow in glaucoma, it's very hard to get patients to keep their eyes still. They have to be able to keep their eyes still essentially for 40 minutes. They're allowed to blink, but the problem is if you make an eye movement, then you have to reacquire that. So we're working on methods of doing alignment, but since the structure is so small, it's very, very difficult to do. Of course, we're going to apply this to glaucoma patients. And overall, I'd just like to say individual variability in visual tactile acuity correlates with cortical magnification. We found that traditional bold MRI is a reliable method for monitoring the progression of glaucoma. Cortical changes in glaucoma correlate with the loss of visual function and retinal nerve fibers. And finally, future plasticity studies will monitor progression of glaucoma in the eye, the LGN, and the primary visual cortex. And hopefully we'll be able to understand this disorder, provide new diagnostic techniques, therapies, and hopefully eventually a cure. I would like to thank the physicians who provided me with patients for this, Dr. Weinreb and Felipe Medeiros at UCSD's Eye Clinic, the Hamilton Glaucoma Center, along with my scientific collaborators, Linda Zangwell, Pam Sample, and Christopher Bowd, and of course MRI collaborators at UCSD's MRI Center for Functional Magnetic Resonance Imaging. Thank you very much. If you have, I guess we have time for questions if you're not exhausted by the massive data. Yes, hi.
Are patients fixating on a fixation point during these retina experiments, or how do you?
Yes, yeah, they have a retinal type. We alternate periods of rest with periods of fixation, so it's not as hard as it initially sounds. They will be allowed to move their eyes at some point in the scanner, but you generally don't want them to because you'll have to reacquire that image. And these are the best of the best. They're very astute graduate students and postdocs. And if we're having problems getting it to work with them, glaucoma patients are going to be much harder. We have to refine the technique. Yes?
Can these applications be used with patients with narrow angle as well as wide angle glaucoma, or are you going to specify one particular type?
Right now we're looking at primary open angle glaucoma, which is the most prevalent form of the disease. It's also the form of the disease that tends to not always accompany increases in pressure. We can look at narrow, we can look at angle closure glaucoma, we can look at everything. But I'm more interested in the cases where there might be a neural or blood-based origin for the disease.
Can this also ultimately be applied to patients with the diabetic neuropathy as well?
Sure. Yeah, sure. The technique can be applied in any realm at this point for the big 4 ocular diseases. If there's something that really damages blood flow, like an edema, it might cause alterations. But we're going to be measuring things relative to that patient population's baseline or intraocular changes. So, yeah. Well, thanks. Yes, Francis.
Any pharmacological treatment for glaucoma?
Well, they have been trying to use methods that treatments to encourage nerve growth, things like memantine that have been used in Alzheimer's. Unfortunately, the first trials were not successful. And right now, if I'm successful in getting this information, we'll be able to distinguish between blood-borne mechanisms and mechanisms that have to do with neurotrophic factors. And if we can sort of divide that camp, then maybe we'll be able to narrow down what types of medicine should be given to the patient.
And there is laser therapy, I believe. So how does that work?
Glaucoma has several types of therapies. Usually they're first given drops, and then there's something called laser iridotomy, where you just drill holes into the eye using a laser, and eventually laser trabeculoplasty, where they actually cut a little flap where the aqueous is supposed to leave. And you can control pressure. If you catch it, you can control pressure and slow down the disease. The key, again, is catching it early, and then, you know, try not to be too aggressive with the surgery, but aggressive enough to stop it in its tracks. Cool. Any others? Well, I guess thank you again for your patience. I'll hang around if you have any other questions.
Provost for sponsoring this. You had a very successful lecture series this year with presentations from the behavioral sciences, the natural sciences, the health sciences, and even the humanities. So we were very comprehensive. The Provost Lecture Committee will meet this Thursday to decide our schedule for the fall 2009 semester. I want to thank the committee members, the Provost, the Assistant Provost, I see Professor Levy, Professor Villegas. We have other members as well. I don't think they're here. Professor Vasquez, Professor Delato. I chair the committee along with Professor Grasso as well, who's a member of the committee. And we have 13 applications for next semester, so we really will have difficult decisions to make. But we're very happy to see such interest in this series, and it's evidence of the vital intellectual life on our campus, and also of the very active research programs pursued by many, many members of the faculty. Without that, we wouldn't be able to have such an exciting series. So thanks to everyone, and I guess Dr. Griffith wants to say a few words.
Two quick words. First of all, thanks to Laura and the committee. Give them a round of applause. And I think it's a tribute, as she said, to the fact that there is not only good instructional activity at York, there is also good discovery, research and discovery activity. And part of that is what we had a taste of today. Some of you may recall that in the fall we started a companion series. A companion series that we're calling Provost's Distinguished Scholars Lectures. This series allows us to see and hear from colleagues within York. The companion series allows us to have conversations with colleagues coming from outside of York who are going to be helping to have us engage in intellectual discourse on a variety of subjects. And what I thought I'd do is share with you the next lecture in the Distinguished Scholars series. If you have a chance, if you're interested in issues of bondage and slavery, come and listen to our colleague from Harvard. Not only is it, I think, a tribute to the interest, what Laura mentioned to us, we've got 13 applications for 3 spots, but it also reflects, and I think we can see the evidence here today, and see the evidence from the other lectures. There is a constituency of students that is involved in coming to these lectures, hearing from some of their faculty mentors, extending their own perimeters of knowledge by being part of the series. What I thought I would do is maybe share with you that next year I'm going to start a new effort to capture the student component. We're going to start an undergraduate student research project Will we allow the students themselves, working with faculty mentors, to showcase their research, to talk about what they're doing in the humanities, to talk about what they're doing in the sciences, talk about what they're doing in the professions, to allow us to be beneficiaries collectively, not only of what the faculty are doing, but what the students are doing. And you'd be amazed how much student stuff, to use my son's favorite term, happens at York. So thanks for being part of this series. I look forward to your not only being part of the Distinguished Scholar Series, but as you get more information in the early fall about the Student Research Project, I welcome faculty and students and staff being intimately involved in that. Assistant Provost, and Coach. Thanks for reminding me. We have a ritual, a tradition, that we give as a memento for the lecture a copy of one of the posters. We frame it and we give it to the lecturer, and this is yours to keep. If you leave York, you got to leave it with us. We frame two of these, and we give one to the lecturer, and we keep one for a gallery, which we're going to be. You would have seen the first year's lectures, that gallery is up. Sometime over the summer, we'll have the second year's lectures gallery up. And as each year goes by, we'll have an additional gallery displaying the posters of the people who were there in that series. But we also didn't have one the last time. Nick gave his talk, and we didn't have Nick's. We had a. Sorry, turn on the projector. We had a make-believe for Nick, so now you get real believe. You're supposed to shake hands with me and look at the camera. And